Hi everyone, I'm Xinyi from the University of Hong Kong Georgia Host Lab. Today I'm gonna present our package we developed recently for differential composition analysis of single cell data. So when we are getting single cell data samples, it's a common occurrence that we will get samples coming from two different conditions, and we are curious about whether the condition will influence the cell type composition. If the proportion we observed from the single cell data can reflect the true biological proportion in the biological sample, it is very natural for us to take the proportion and doing the analysis based on it. However, during some because of some technical noise and some noise introducing during the clustering process, and this might be a problem. For example, if we look at the UMAP here, we can find out that this CD8 T cell has some part overlap with other cell type, and the right matrix here is the misclassification matrix we estimating, showing that one cell type might be misclassified to another cell type. And in this case, we can find out that this CD8 cell only have 0.8% being correctly assigned to itself. And in some cases, it will be misclassified into another cell type, and this introduces a misclassification error. So our package based on two assumptions. The first one is that cell counts follow a beta-binomial distribution. And the second assumption is that this kind of misclassification error exists and can be somehow described by the misclassification matrix showing here. And this is the basic workflow of DCATS. The three matrix highlighted with light blue here. Count matrix, design matrix, and misclassification matrix is three input required by DCATS. The count matrix indicating the number of cells for each cell type in each samples. And the column name here is the cell type, and row name represent the sample. And the design matrix is the information about those the experimental design that might influence the cell type proportion for each sample. And the misclassification matrix is the matrix I described before to describe to showing the misclassification error. With the help of misclassification matrix, we can use expectation maximization to estimate a latent true matrix. And for each cell type, we can fit a beta-binomial model, GLM model, adding up with likelihood ratio test to get the final result, which is the p-value, helping us to decide whether a covariate will influence the cell type proportion. And in the DCAT workflow, there are three parts that seem very important. The first is how to estimate this misclassification matrix. And second is the chosen between the base model, full model and non-model. And third is whether to use this fixed over-dispersion term. DCATS supports three ways to calculate a misclassification matrix. The first is a uniform typed matrix. In here, we assume that for one cell type, the probability it being correctly assigned to itself equals to A, and the probability for this cell type being assigned to the other rest cell type equal to 1 is the same, which equal to 1 minus A divided by K minus 1, and K here indicating the number of cell type. And for the second, Kn type matrix is estimated from the Kn graph, be calculated by the proportion of cell type I's neighborhood that belongs to cell type J. So if we want to calculate the probability for cell type I being assigned to cell type I itself, I and J should be equal here. And the third is a SVM type matrix, where we require a cell type label and other information that user believe will influence the clustering result, combined with a data set. And we will split this data set into 5 parts. For each part, for each time, we will select 4 of them to build to train as a training data to train a support vector machine, and then predict the cell type label for the rest part. By repeating this process 5 times, we can get a predicted cell type label. And by comparing it with the original cell type label, we will get the misclassification matrix. And for each cell type, we will fit beta-binomial GLM model. And this requires a cell count vector from Y1 to Ym, which and Y1 indicating the number of cell count for each cell type in the first sample, and so on. And the design matrix describes the experimental design information for sample 1 to sample m. And if we use mu to denote the mean of y, then we can use a logic link function to link the mu with a linear combination of the experimental design information x1 to xn and get a beta-binomial GIM model. And in here, as we using a design matrix, so actually it allows us to include not only the condition information, but multiple covariates at the same time. It might include age, sex, sample type, and condition. And here we will have 2 types of model to do the beta-binomial GLM part. The first non-model, we only use the covariate we want to test it each time. So we will comparing the model 0, which do not include the tested covariate information, and model 1, which will include the tested covariate information. By comparing the model 0 and model 1, we can see by using the likelihood ratio test, we can see whether adding up this tested covariate information will help improve the fitness of the whole model. And in other words, we can see whether this tested covariate will influence the cell type proportion. And if we choose the full model, except for the tested covariate, in both model 0 and model 1, we will add the information of other covariate except for this tested covariate. And in this case, we are actually testing the influence of the tested covariate while controlling the influence of other covariates. As I mentioned before, we assume that cell counts follow a beta-binomial distribution, which means that we should also we also need to estimate a over-dispersion term phi. Normally, when we are fitting different beta-binomial GIM for each cell type, the phi is flexible for each GIM will have its own phi. However, in some cases, we might have only 2 or 3 biological replicates, and the estimation of phi will be not that accurate. It might occur as an overestimation problem. And in this case, we will borrow the information from other cell type and estimate a global phi across all cell type before using the GIM to do the following testing, and use this phi as the fixed phi term in the GIM model to do to calculate the p-value. This is what a count matrix looks like. So the column name here is the cell type, and the row name is the sample name. 36, for example, 36 here indicating that we observe 36 cells that belongs to the endocrine group that in the sample B1. And here we have 6 samples. And for the design matrix, we assume that in this case, we assume that only the condition group will influence the cell type proportion. And the 4 samples coming from the control group, and the last 2 coming from the H poly 3 group. So that's why this design matrix look like this. And we also support a design matrix with multiple columns, and in this case, we might include more information. A misclassification matrix showing the misclassification error. For example, the 0.979 here indicating the cell type endocrine being assigned to itself, the probability is 0.979, and the sum of each row should be equal to 1. As I mentioned before, we have 3 ways to estimate the misclassification matrix. So here, we use the following 3 functions. The first is estimating a uniform typed misclassification matrix. So the input including a cell type number and a confusion rate. And confusion rate here indicating the probability of one cell type being assigned to other cell type, but not itself, is the confusion rate. And the KN type misclassification matrix require the input of an KN graph, which is this, which here we use the KN graph in the SARET object. And also a simulation, a cell type label based on the clustering and annotation result. And this SVM type require a data frame. And in this data frame, it should include a cell type label, getting from the annotation result, as well as other information that can be, that the user think will influence the clustering process. And here comes to the main function of DCATS. The main function of DCATS is this DCATS GRM function. And it requires several input. So 3 basic inputs should be the count matrix, the design matrix, and the misclassification matrix. And here we assume that this is the count matrix and this is the design matrix. And in default, DCATS GRM used the non-model, and it will estimate a different over-dispersion term phi for each GRM model. But if we want to change it from non-model to a full model, then we can specify this base model term equal to 4. And if we want to use a global estimated phi for this DCATS GRM function, we can first use this get phi function to estimate a global phi, and then we can use it as the input for this DCATS GRM to get the final result. And this is the main result for this DCATS GRM function, and it is the p-value of the likelihood ratio test. So the null hypothesis here is that for each cell type, the proportion of samples coming from different covariates will not be influenced. So here as 0.8939, this part is larger than 0.05, we can get the conclusion that for cell type 1, its proportion in samples that coming from different condition group is the same. To demonstrate to evaluate the performance of DCATS, here we design this simulation process. First, we generate a very large cell pool from the library splatter, which include gene expression matrix, true label, and we will define a proportion vector for sample coming from each condition, and using a directional distribution adding up with a multinomial distribution to get the final cell count, to simulate a final cell count for each cell type. Based on the cell count, we will select different cells from the cell pool we generated before and combine it with the simulated data, and using Seret to do the clustering process and get the count matrix, design matrix, and misclassification matrix to do the differential composition analysis. And we compare DCATS with other four methods, where Speckle is an unpublished package doing the same thing. And SCDC is a published package doing the same thing, while DiffSci is a published package doing differential composition analysis for flow cytometry data. In order to exclude noise coming from the clustering result, we also use a theoretical simulation process in which we only simulate count matrix, design matrix based on a misclassification matrix we define and do the differential composition analysis. Here is the result. So for the theoretical simulation, we simulate design matrix and we simulate count matrix based on this misclassification matrix, and you tested both on DCATS and Fisher. If we do not use this true similarity matrix to do a correction for the misclassification error, we will observe a relatively lower AUC. While using this misclassification matrix to do this correction on the misclassification error, we can observe a significantly increase on this AUC, which showing that with the help of a true misclassification matrix, this differential composition differential testing will show a higher accuracy. And we using this whole simulation design to explore the performance of DCATS in different scenarios. So we changed the number of biological replicate in each condition from 2 to 4, and changed the number of cell type from 8 to 12. And we can see in all the situations, DCATS have the highest AUC value. And if we look into detail about MCC sensitivity and specificity, we can find out that even though the Fisher and SCDC method have a relatively higher sensitivity, their specificity is very low. And in the real world scenario, we find out that they will give lots of false positive results, which will influence their final result, the final accuracy. And to test the function of multi-covariate, we also adding up the influence of age and gender. And DCATS not only shows a high performance comparing to other four methods, it can also give the estimate the influence of 3 covariates simultaneously, while other four methods did not support this function. And we also test DCATS in different real-world data. This first data is a PBMC single-cell RNA-seq data, where they have 2 conditions, stimulated group and unstimulated group. And based on the original paper, the stimulation itself won't influence the cell type proportion. So if we do the differential composition testing, we should get a non-significant result for all cell type, and this is what this is the result DCATS gave us. But for the Fisher and SCDC method, they will give some false positive result. And this is another real-world data where we have 4 conditions: control group and infection for samples coming from different days infection of different bacteria. Similar to the previous real-world data, Fisher and SCDC will give some false positive result, while speckle is not that sensitive. It gives almost non-significant. NS here represent non-significant, and it gives almost lots of non-significant result. Even for those cell type that should be differential, that should have different proportions. And we also test DCATS in COVID samples coming from COVID-19 patients and its corresponding control group. In the original paper, when they are doing the differential abundance analysis, they are using only the condition information, which means they are only using which group this patient in. However, as DCATS support multi-covariate, we adding up other meta-information, including age, gender, and doing the analysis. By controlling the information of age and gender, we observed another, so the significant bar colored with black are the result consistent with the original paper. And except for this finding, we also find another pair of cluster that shows differential abundance, which is not found in the original paper. And if we look at it clearly, we can find out that this new finding came from the group comparing the control group and the mild group. And we can observe the same trend when comparing the control group with the severe group. So we believe that this finding is reasonable and might be masked due to the noise of age and gender in the original paper. And this is what I'm sharing today. And I would like to thank you for the lab, Joshua Ho's lab and Yuanhua Huang's lab in the Hong Kong University of Hong Kong and all the lab members.
Thank you. Reminder again to everyone, if you want to ask the speaker a question, please use the Slack channel. I have a question myself, although I'm not by any means an expert in this field or single cell data at all. But would you please comment on like the resources required to perform the differential expression, the differential composition analysis using DCATS?
Sorry?
The resources required to run this kind of analysis.
3 matrix. You mean the input of this kind of analysis?
I mean the computational resources to run this kind of modeling.
Oh, it's an R package, so yeah,
okay.
Can simply install it from GitHub, and we were considering like adding up, submit it to bioconductor after we like polish it.
Okay. We have a question here from Belinda. So when you compare the speckle, I wondered if you use the arcsine square root transform or the logarithmic transform.
So I'm not sure which part it infers to, but if it infers to the expression matrix, which is commonly in the single cell RNA-seq data, we do not require that matrix. We only require the number of cell count and experimental design information for each sample and a misclassification matrix. Yeah, so for the gene expression matrix in the single cell RNA-seq data, we don't have a specific requirement on it.
Okay. I would like to thank you again. And again, if you like to get in touch with the speaker, please use the Slack channel. And in a few minutes, we'll be back with the last talk of today. Thank you.
Thank you.